Yes.
Yes, it's open. It's Lilia.
Okay.
However, for the purpose of the project, I decided to talk a bit. You can. Well, I wanted to cover all the process of development of a serological assay, starting from the preparation of antibodies, because that is a part of our project. So, of course, I'm not going to explain you how ELISA test works, because otherwise I will bring you back to the past. But I think it's more important to understand, to point out the key issues and the critical point in the developing of a polyclonal antibody. So, besides a brief introduction, I will go to the description of the process. First of all, I would like, as Tanya already mentioned at the beginning, in her presentation, I would like to focus on where we are when we use the serological test. Generally, the ELISA test, which is the gold, the standard gold of immunoassays, applies to large detection when we have to screen a large number of samples, or when we need a double check, especially when we deal with regulated pathogens. And this is the case where we are in the TROPICSAFE. When a diagnostic test has a supposed to apply in regulated pathogen, it has to meet some technical requirements, and so I wanted to point you out the ECHO standards number 76 fourth version and 98 second version, where we can find how to validate the requirements to validate a serological, well, not in general, any diagnostic tool. We have just a quick glance to an example of Xylella fastidiosa. We were involved in a European project, and we can see that the serological test, here are ELISA test and DTBR, integrated LAMP and PCR. So they move together in the first step, basically the first step, and also when you have to monitor in the outbreaks area. Then just a few, Wikipedia keywords. When we talk in a serological test, when we talk about epitope, we talk about no, sorry.
Effect. La freccia, una delle due di lato.
No?
Quell'altra.
Ok.
Yes.
No.
Freccia avanti, freccia
I do it.
Okay. Yeah. When we talk about an epitope, we refer to a target. Antigen or epitope or antigen, we refer to the analyte, the target of the detection. When we talk about the analytical reagent, we refer to an antibody, and its quality is basically measured and assessed by the affinity. And when we talk about the outcome of the assay, we refer to the immunocomplex, and particularly immunocomplex stability gives us the quality of the system, of the analytical system. So we have the antigen or epitope, we have the antibody as the reagent, and then we have a very simple okay. So this is the antigen, this is the binding site between the antibody and the antigen. It's very simple compared to the lab description. So now back to the project. As I said, we will talk specifically about polyclonal antibodies. So we are not talking about monoclonal or recombinant single chain recombinant antibodies. And we wanted to develop specifically ELISA or lateral flow techniques for LY and AY. And then we go further to validate the test. So the process, which is a development and production process, we can break it down in three stages. Well, of course, we have a feasibility study before getting in a project of development. And then we needed to prepare the antigen, we needed to rise the antisera, and then we choose the technique. The antisera are, I don't know, maybe some are produced in animals. So the most commonly used animal for polyclonal antisera is the rabbit, and which works here like a bioreactor. I call them the bioreactors. And the crucial part is the preparation of the antigen, and in this phase we are linked with the work of the cultivation of phytoplasma. And so the link between the Nicoletta work and our work is the first stage is the preparation of the antigen. So, okay, this is something about the feasibility study. I go faster here. So we know that we are aware that polyclonal and monoclonal antibodies were developed in the past. However, so far no serological tests are available to detect due to the poor specificity. Pure specificity.
I do. This, I do it. When you have to go back, tell me.
Thank you. So what is new? The new challenge is that we have available two new technologies. One is the cultivation of phytoplasma, like Nicoletta described before, and then we have advanced studies in the molecular studies in the phytoplasma membrane proteins, and ELISA will give a presentation after that. So when we prepare an antigen, we work on three parameters. Is the quantity. We work on how much, the amount, the identity, and the purity. These are three fundamental parameters we need to assess. The quantity has to be sufficient to implement the immunization program, and that depends on the antigen source. If we have infected plants, culture, or recombinant proteins. The identity is important because it determines the antibody specificity, and then also depends on the nature of the target. And then we have the purity of the antigen preparation, which again generates the specificity and the affinity of the antibody. Okay. So when we'll be focused on bacteria now, and later also in recombinant proteins. How do we prepare the antigen preparation? When I say antigen preparation, I mean what the preparation we use to immunize the animal. So it has to be injected in the animal. We know that with bacteria, while differently from viruses and recombinant protein, we deal with one single, by and large with one single substance, which is the code protein, for instance, in the virus. When with bacteria, we know that we have a mix of substances. So that means that the purity assessment is not carried out directly on the mixture of substance, but it depends on the bacteria culture. That is something that we have talked with Nicoletta. So generally here, when we talk, we deal with viruses, the limitation is the amount we get from a virus purification. The bacteria, we have low identity of the antigenic part, very difficult, almost impossible to define the purity. And while when we deal with the recombinant proteins, we have the benefit of non-limited production, but we don't know how much identity there is between the recombinant protein and the target of the analysis. Okay, here the way we assess the requirements of the antigen preparations. Here we are with bacteria. We simply express the amount and the purity and altogether by the number of CF2 or by OD. Well, actually, we asked also Assunta if it's possible to use the microscope, the electronic microscope, to assess, to look at the bacteria cultivation. Okay, now we rise Antisera, and what is important in terms of processing is that the immune response is not predictable. So once we have injected in the animal, we don't have a control of the process. So that's why it's so important to have an optimal antigen preparation. Well, once we obtain the antisera, the first quality assessment of the antisera consists in titration to prove specificity, titration against the antigen, and to prove the specificity and the affinity of the antibodies to the antigen. Then if we want, we can evaluate the antisera in Western blot, immunoscopy, microscopy, and immunofluorescence, and for bacteria. And it is important also to investigate on non-specific reactions, because at this stage we can take sure to improve the quality of the antisera, and which consists basically in the pre-absorption against the contaminants. Okay. And then this is just a consideration that once we have the antibodies and they pass the first checkpoint, which is the specificity and the affinity, then the application of the antibodies to the different immunotechniques is something that has to be proven. It cannot be predicted. I mean, if an antibody works in ELISA, that doesn't mean that would work in other immunoassays. So here the two techniques we have chosen in our project is the ELISA and the lateral flow. When we assess, when we deal with an ELISA test, basically the development process consists in the development of the assay protocol. So the protocol is the core of the assay, of the development. When we deal with the lateral flow, the crucial, the key issue of the development is how we can incorporate and distribute the antibodies on the device material, which are the membrane, the pad and the membrane, and the cellulose membrane. So just to compare the intended use, as we said, between to compare the two techniques, as we said before, ELISA is for large test numbers, the lateral flow is to intercept the pathogen. The ELISA is suitable to meet standard operation procedures for the accreditation of testing laboratories, while the lateral flow is suitable for field tests. ELISA no rapid lateral, okay, we know that. And about the cost, the ELISA, the cost per test is definitely lower than the lateral flow. So far for plant pathogens. I mean for the medical field. By definition, the ELISA test is a solid phase surface assay, and the detection signal develops by a chemiluminescent reaction, and the results can be interpreted not only qualitative but also in a semi-quantitative way. Okay, yes, what is when we have an ELISA, what we have to produce? We have to produce the capture reagent, the conjugate reagent. We have to evaluate the ELISA plate. We have to assess the protocol, and we have to figure out the cutoff, which is the test to be used to discriminate the outcomes. Well, this is what we do. Technically, it's IgG purification, high affinity chromatography, and conjugate the IgG with the enzyme, which generally for plant pathogen is the alkaline phosphatase. How to decide? Well, I go fast on this subject because it's just a description. But what I want to point out for the benefit of the project is where we add value when we develop a test, the assay. In our experience, the crucial point is the calibration of the test. And so how we conceive our reference material against which we calibrate the reagents. And most important is how to standardize lot by lot production. And how we enhance the quality of the diagnostic tool as it is perceived by the end user. Generally, the end users prefer short protocols. They want to know if we are using polyclonal or monoclonal antibodies because it has to do with the specificity of the test and the variability of the target pathogen. And of course, the lowest background and the easy cutoff assessment. And also, it's important to have available the positive and negative control of the assays so they can carry out their own independent quality control and validate the results. So now the lateral flow. The lateral flow is by definition an immunochromatography process. It's developed the signal, the detection of the signal is developed by a colorimetric reaction. And what is very important here is that the affinity is the quality of the antibodies. Again, the high affinity and high concentration are crucial to build the device. And then, of course, you know that the outcomes are strictly qualitative. The technical, the components of the lateral flow are basically a device, the plastic cassette which contains the device, and then we have the reagents. Here, okay, you can find the Wikipedia how it is constructed, the components. I don't know if you like it, we can describe how it works, but I think we have no time. Let me see what I wanted just to, maybe we have no time, I want to finalize the lateral flow and the limitation. The limitation there are some aspects. One is the sensitivity of the assay. We cannot load the sample with high volume preparation. We cannot we have to load the device with a small amount of the samples. And then the process of drying and re-grading the antibody on the membrane is very critical for the maintenance of the antibody reactivity to the target. And then the construction, the preparation of the device requires a very accurate application of by instrumentation, which is very expensive, is a high investment equipment. Generally, it's a process that we can externalize to professional other companies. And well, what I wanted to stress is that to construct a lateral flow, we need a large amount of antibodies. Just to give you a point of references, with the amount we have to place in one test, a lateral flow test, we can carry out about even sometimes more, 10,000 tests, ELISA tests. So there is an extreme, I wouldn't say fast of antibodies, but I mean the amount of antibodies. So that's why the lateral flow is mainly prepared by monoclonal antibodies. Because the production of monoclonal antibodies is unlimited. But on the other hand, the specificity can be, the range of the specificity too narrow. And so that's all. I had just, the conclusion is the open questions, because I was required to pose questions for the debate, that I think we're not going to do. And so it was about if preparing the antigenic preparation of from phytoplasma can be followed the same procedures we use for bacteria culture, and there was something about the recombinant proteins, if we can use a recombinant protein derived from phytoplasma, to be used not only to prepare an antibody against that, but also to be used as a reference material in ELISA. And then about the lateral flow was about the map, the use of polyclonal or monoclonal antibodies.
So actually, I will only speak about the possibility of using a recombinant protein coming from the membrane protein of phytoplasm. Actually, with Lilia and with the Coletta, we thought to use two approaches to produce antibodies that will be used then to develop a lateral flow or whatever to use. The first, as you heard in Coletta, the use of the whole prokaryotic cultivation as antigen. And obviously, we were discussing, yesterday we did, and today we will do it again, if some cytoplasma are available, because some are not yet available, if it's possible, the purity, the purification, and so on. The second is another different strategy, but with the same aim. It's the use of the recombinant membrane protein of the prokaryotes as antigen. So we use one protein, but to have this protein, we need to identify the gene sequence. And we have to check if we have it or if it's available in gene bank, and then we have to express the protein in an Escherichia coli medium. Not a medium. Sorry. Okay, so I will speak only of this part. And as you know, the phytoplasmas has a subset of membrane proteins that have these antigenic properties, immunodominant properties. They are called immunodominant membrane proteins. And they constitute generally the major part of the membrane proteins in most phytoplasmas. We have three types of these proteins, which are not homologous among them. And they are in fact very different, we will see in a few seconds. The first type is the IMP, immunodominant membrane protein. The second is IDPA, immunodominant membrane protein A. And the third is AMP, antigenic membrane protein. That looks very similar, but are different in a way. So this is the shape, you see? IMP, IDP, AMP. Probably the function is the same, but they develop independently in different phytoplasmas. So here is the outside of the cell of the phytoplasmas, here it is inside. So we have a transmembrane domain or two, all of the three. And then this is the immunodominant portion. We are interested in this, or this. So, as I told you, and I didn't tell, generally a phytoplasma, one species, one phytoplasma, has one dominant membrane protein, so one of the three, but some of them have two. One is the major and one is the minor. And they are very divergent because they are subjected to very strong divergent positive selective pressure. And they also are located in different parts of the genome. So, for example, the IMP is between the DND and the PFG. The EDP is after the DNA X, and the AMP is between the GROL and the NADP gene. So you see just from here that they are different genes. And here the most important, because actually this table was not complete, and I could complete this yesterday with the discussion that we had with the groups. So our main target in GRAPEVINE could be, among brackets you see the name which is not official, and maybe will never be, but is the Phytoplasma flavescens, the fifth group, which is the protein is the IMP. And yes, we have the sequence. There are not, one is published from Torino, but the other we have most of them, 40 of them in our here at CREA. Candidatus Phytoplasma solani, which is in EU and also in Europe and sometimes in Chile, has the AMP protein, which is in gene bank actually. A lot of them, something in French sequence, I think 40, 50 of them, a lot. Then we have the Candidatus Phytoplasma asteris, that is mainly our target in South Africa. And the main protein here is AMP, even if it should be also the IMP. And a few sequences are available in gene bank. Not exactly of the phytoplasma present in South Africa, but of other strains. And then the 3J phytoplasma of GRAPEVINE in Chile, which has the other type. So with the 4 phytoplasmas, we have the 3 types of protein, the EDP. And actually, the paper was just published from Nicola Fiore, and we have the sequence exactly of the EDP in here. So this is what we have. For palm, as we heard yesterday, we have the phytoplasma palmicola, which is in Africa, has IMP protein, and Matt told me that together with Ghana they had this sequence, so that's good. Then for the 4 group that I didn't understand well which subgroups were, sorry Carlos, but is it right because I was not sure. So the protein, anybody knows which one is, and if there is one. So there are no sequences available, and probably I think that if we want to think to develop something from these strategies, this is the less indicated, obviously, because there is a lot of work to find out the sequence, the protein, and then obviously you don't need only one sequence, you need more than one, because you want to make a protein which is representative or the genetic variability you have in your field. So this is our starting point, actually, and at the beginning I was thinking to discuss all of this with you, but for the discussion of yesterday I got all this information. And then this is Candidato di Peribacter, I mean, this is, for me, is a question point,